In molecular cloning, a vector is a DNA molecule used as a vehicle to artificially carry foreign genetic material into another cell, where it can be replicated and/or expressed. A vector containing foreign DNA is termed recombinant DNA. The four major types of vectors are plasmids, viral vectors, cosmids, and artificial chromosomes. Of these, the most commonly used vectors are plasmids. Common to all engineered vectors are an origin of replication, a multicloning site, and a selectable marker. The vector itself is generally a DNA sequence that consists of an insert and a larger sequence that serves as the backbone of the vector. The purpose of a vector which transfers genetic information to another cell is typically to isolate, multiply, or express the insert in the target cell. Vectors called expression vectors specifically are for the expression of the transgene in the target cell, and generally have a promoter sequence that drives expression of the transgene. Simpler vectors called transcription vectors are only capable of being transcribed but not translated. They can be replicated in a target cell but not expressed, unlike expression vectors. Transcription vectors are used to amplify their insert. Insertion of a vector into the target cell is usually called transformation for bacterial cells, transfection for eukaryotic cells, although insertion of a viral vector is often called transduction. Characteristics Plasmids Plasmids are double-stranded and generally circular DNA sequences that are capable of automatically replicating in a host cell. Plasmid vectors minimalistically consist of an origin of replication that allows for semi-independent replication of the plasmid in the host. Plasmids are found widely in many bacteria, for example in Escherichia coli, but may also be found in a few eukaryotes, for example in yeasts such as Saccharomyces cerevisiae. Bacterial plasmids may be conjugative transmissible and non-conjugative. Conjugative mediate DNA transfer through conjugation and therefore spread rapidly among the bacterial cells of a population. For example, F. plasmid, many are and some col plasmids, non-conjugative do not mediate DNA through conjugation, for example, many are and col plasmids. Plasmids with specially constructed features are commonly used in laboratory for cloning purposes. These plasmid are generally non-conjugative but may have many more features, notably a multiple cloning site, where multiple restriction enzyme cleavage sites allow for the insertion of a transgene insert. The bacteria containing the plasmids can generate millions of copies of the vector within the bacteria in hours, and the amplified vectors can be extracted from the bacteria for further manipulation. Plasmids may be used specifically as transcription vectors and such plasmids may lack crucial sequences for protein expression. Plasmids used for protein expression, called expression vectors, would include elements for translation of protein, such as a ribosome binding site, start and stop codons. Viral vectors. Viral vectors are generally genetically engineered viruses carrying modified viral DNA or RNA that has been rendered non-infectious, but still contain viral promoters and also the transgene, thus allowing for translation of the transgene through a viral promoter. However, because viral vectors frequently are lacking infectious sequences, they require helper viruses or packaging lines for large-scale transfection. Viral vectors are often designed for permanent incorporation of the insert into the host genome, and thus leave distinct genetic markers in the host genome after incorporating the transgene. For example, retroviruses leave a characteristic retroviral integration pattern after insertion that is detectable and indicates that the viral vector has incorporated into the host genome. Transcription is a necessary component in all vectors. The premise of a vector is to multiply the insert. Thus, even stable expression is determined by stable transcription, which generally depends on promoters in the vector. However, expression vectors have a variety of expression patterns, constitutive or inducible. This expression is based on different promoter activities, not post-transcriptional activities. Thus, these two different types of expression vectors depend on different types of promoters. Viral promoters are often used for constitutive expression in plasmids and in viral vectors because they normally force constant transcription in many cell lines and types reliably. Inducible expression depends on promoters that respond to the induction conditions. For example, the marine mammary tumor virus promoter only initiates transcription after dexamethasone application, and the Drosophila heat shock promoter only initiates after high temperatures. Expression vectors produce proteins through the transcription of the vector's insert followed by translation of the mRNA produced. They therefore require more components than the simpler transcription-only vectors. Expression in different host organism would require different elements, although they share similar requirements, for example a promoter for initiation of transcription, a ribosomal binding site for translation initiation, and termination signals. Prokaryotes expression vector equals, promoter. Commonly used inducible promoters are promoters derived from lac operon and the T7 promoter. Other strong promoters used include TRP promoter and TAC promoter, which are hybrid of both the TRP and lac operon promoters. Ribosome binding site follows the promoter, and promotes efficient translation of the protein of interest. Translation initiation site, shined alganose sequence enclosed in the RBS, 8 base pairs upstream of the August start codon. Eukaryotes expression vector equals, eukaryote expression vectors require sequences that encode for polyadenylation tail, creates a polyadenylation tail at the end of the transcribed pre-mRNA that protects the mRNA from exonucleases and ensures transcriptional and translational termination, stabilizes mRNA production. Minimal UTR length. UTRs contain specific characteristics that may impede transcription or translation, and thus the shortest UTRs or none at all are encoded for inoptimal expression vectors. Kozak sequence. Vectors should encode for a Kozak sequence in the mRNA, which assembles the ribosome for translation of the mRNA. Features. Modern artificially constructed vectors contain essential components as well as other additional features. Origin of replication, necessary for the replication and maintenance of the vector in the host cell. Promoter. Promoters are used to drive the transcription of the vector's transgene as well as the other genes in the vector such as the antibiotic resistance gene. Some cloning vectors need not have a promoter for the cloned insert, but it is an essential component of expression vectors so that the clone product may be expressed. Cloning site. This may be a multiple cloning site or other features that allow for the insertion of foreign DNA into the vector through ligation. Genetic markers. Genetic markers for viral vectors allow for confirmation that the vector has integrated with the host genomic DNA. Antibiotic resistance. Vectors with antibiotic resistance open reading frames allow for survival of cells that have taken up the vector and growth media containing antibiotics through antibiotic selection. Epitope. Vector contains a sequence for a specific epitope that is incorporated into the expressed protein. Allows for antibody identification of cells expressing the target protein. Reporter genes. Some vectors may contain a reporter gene that allow for identification of plasmid that contains inserted DNA sequence. An example is LAC-ZI+, which codes for the N-terminus fragment of I-squared galactosidase, an enzyme that digests galactose. A multiple cloning site is located within LAC-ZI+, and an insert successfully ligated into the vector will disrupt the gene sequence, resulting in an inactive I-squared galactosidase. Cells containing vector with an insert may be identified using blue-white selection by growing cells in media containing an analog of galactose. Cells expressing I-squared galactosidase appear as blue colonies. White colonies would be selected as those that may contain an insert. Other commonly used reporters include green fluorescent protein and luciferase. Targeting sequence. Expression vectors may include encoding for a targeting sequence in the finished protein that directs the expressed protein to a specific organelle in the cell or specific locations such as the periplasmic space of bacteria. Protein purification tags. Some expression vectors include proteins or peptide sequences that allows for easier purification of the expressed protein. Examples include polyhistidine tag, glutathione S-transferase, and maltase binding protein. Some of these tags may also allow for increased solubility of the target protein. The target protein is fused to the protein tag, but a protease cleavage site positioned in the polypeptide linker region between the protein and the tag allows the tag to be removed later. See also, plasmid, viral vector, cloning vector, expression vector, minicircle, recombinant DNA, naked DNA, vector, an organism that transmits disease. References: Freshney, Ian. A Culture of Animal Cells, A Manual of Basic Technique. John Wiley & Sons, Incorporated, Hoboken, New Jersey. ISBN 978-0-471-45329-1. External links: Waxman Scholars Introduction to Vectors, A Comparison of Vectors in Use for Clinical Gene Transfer, Gene Transport Unit,